To make silk solution in the lab, we start with an abundance of cocoons. Following removal of the worm, we boil off the coating protein sericin and then dissolve the intact fibers. Following desalting, a solution of silk and water results, which can then be applied to biological specimens. In this example, we see that the volume of a finger prick can be added to the silk and dried down to a thin film, which is mechanically robust for purposes of transport. The entrapped biologics remain conformationally stable. We simply add water to free up the analytes for detection. To test this approach, we gave films of silk-stabilized blood and plasma to a clinical lab for processing and asked them to run a cardiovascular protein assay. We found that both plasma samples in white and blood samples in black performed the same as the liquid samples in gray, but for certain markers, the liquid samples decayed at elevated temperatures. Interestingly, for certain markers, the dotted line indicates damage from freeze-thaw, which we can avoid using the silk system. We are not limited to proteins, and so can consider a number of different markers of interest for an array of disease cases.